All right, now it's time to start talking about the development of the bird embryo. I'll often say chick embryo. Usually the chicken was used as the example of a bird embryo type. Simple enough. But anyway, the bird embryo is going to look quite different than the frog embryo. Remember that the process of gastrulation in a amphioxus, first of all, was just a caving in of cells at one end to form a tube. In the frog, we had this odd folding under of cells that eventually looped over the yolk toward the other end to form the gut tube. So it's a little bit different in that process of early development of the gut tube there. The bird is going to be even more different than the other two in the formation of the gut tube. The bird, when it starts out, the single cell stage, there's a tiny nucleus and there's a big old yolked egg there. So all the yellow of your chicken egg is all this part down here, and on the very top of it, there's a tiny nucleus in all of that mass. That's the zygote stage right there, the single cell stage, and it's going to become a two-cell stage, and when it becomes two-cell stage, there are two tiny nuclei divided by a tiny little cleavage furrow right there at the top. So a very, very small separation of cells going on here. And then when you get to the next stage, again, you're going to see tiny cleavage furrows dividing up the top of that egg. And that just keeps going on. So these are cleavage stages, two-cell stage, four-cell stage, and so on, and eventually we'll get to a condition in the bird egg where we have a number of cells in a layer developing at one end of the yolk. So you'll just have a solid layer of cells, and we refer to that region on a bird egg where the cells are dividing there as the blastodisc. Blastodisc, where those cells form just a little circular pattern of cells right over the yolk. So we've got all the yolk down here and just a single layer of small cells there called the blastodisc. Eventually that region on the blastodisc will form a larger region, slightly larger region, and at the same time separate into a layer of upper cells and lower cells. The upper layer of cells we're going to call epiblast cells. The lower level is going to be hypoblast cells. So rather than the formation of a cavity within a sphere of cells like we saw with amphioxus and frog embryos, here we have the formation of a cavity by what we call delamination, separating layers of cells in the chicken embryo. So this would be the beginning of a blastula stage, where we have a cavity in between a layer of epiblast cells and hypoblast cells. So those above are epiblasts. Those below are hypoblast cells. A two-layered embryo. Now, like I say, this forms a little circular spot on one end of the yolk. Next thing we're going to do is we're going to look down at that circular spot from above to see the next events in the developmental process of the chicken egg. I'm going to use blue here to represent the upper layer of cells, the one that you can see from the top. Now underneath of that, deep to that, you're going to have that cavity, and then beneath the cavity you have the hypoblast cells, and I'll indicate that with an orange dotted line going around here for now. Okay, so think of this as looking down on a sandwich where the upper layer is the epiblast layer, then you have a cavity, then you have that layer of hypoblast cells represented by the orange dot there. some layers of cells. In this upper layer of cells of the blastodisc that we have here, we're going to have a thickening of cells begin at the posterior end of this and extend forward. So it's just pretty short to begin with. This is the beginning of what we call the primitive streak. You didn't have a primitive streak in a frog or an Amphioxus embryo. I'll just put this in here to remind us that this orange part in here, the dotted line, represents hypoblasts layer. Hypoblast layer, and on top in the blue we have the epiblast layer. So we have the two-layered embryo, and in the epiblast, in the top layer, what's going to be our ectodermal layer, we have the beginning of the primitive streak. In our next picture here, we'll have basically the same thing, but the primitive streak is going to enlarge and extend forward across the surface of that blastodisc there. So our primitive streak has expanded, and at the very anterior end of that primitive streak, right there, a little divot forms, a little depression in the very end of it. That little divot or depression sometimes called the primitive pit. The area surrounding that little divot right there is referred to as Henson's node. So Henson's node with primitive pit, and the remainder of this back here is primitive streak. Primitive streak back behind. We still have our layer of hypoblast cells below this layer of ectoderm we're looking at here. From the primitive pit and Henson's node, we're going to get a spreading of mesoderm. So mesoderm in red here is going to spread out from that point to fill that space between the surface ectoderm and the hypoblast cells we see there. Which brings us to our next picture. Okay, now we still have the mesoderm spreading out, but it started spreading from up here in the front. So now we have a field of mesoderm spreading out around this embryo, shaped something like this, so that it's moved out from the Henson's node location, spreading out through there. Now, this end point of infolding of mesoderm started up in here and started spreading that mesoderm out, and it's continuing as it goes backwards. This is kind of moving backwards, so I always think of it as kind of like a zipper. So things are closing in, and as it closes in, as it moves backwards, this primitive pit, remember, the primitive streak started growing from behind and extended upward. Now it's going to regress and go back the other way, and it's laying down mesoderm as it regresses and moves backward through the ectoderm on the surface of this thing. In front of where in front of the Henson's node, where we used to have the Henson's node, is left a thickened zone of ectoderm, so this is the beginning of our neural plate. In red, we have our spreading mesoderm. Spreading mesoderm layer, and of course under that we have our hypoblast layer. If we were to make a cross section through this portion of the chicken embryo right here, let's make a dotted line through there. And we were to see what that looked like in a cross-sectional view, we would have our surface layer of ectoderm going across here that would be thicker in the zone where the neural plate is. So we have a thick neural plate and we have surface ectoderm there. And then we have mesoderm spreading out to either side . Remember, it starts in the middle and it moves laterally, so this is all going out to either side. Then we have below that our hypoblast layer, still in there. There's our hypoblast cells. Some of the cells that it folded in along the primitive streak as it regressed, that were forming mostly mesodermal tissue, some of that will also form some of our endoderm of our gut. And it's going to come right up here and kind of incorporate along that hypoblast layer of cells. So some of our gut is from hypoblast possibly, and then some of it from cells that folded in with the mesoderm as the primitive streak moved back. So this is nothing like what we saw in the Amphioxus or the frog, this primitive streak formation and the infolding of the germ layers here. So we still don't really have a digestive tube, but we're forming some endodermal tissues from this lower layer here. We're going to get a notochord to form right in the middle of all this under the neural tube as it forms in there. Let's draw another illustration of the tissues that are right here in the middle of Henson's node going through that primitive pit. In that zone there, things look more like this. You have this very thin layer of cells, the ectodermal cells that fold in right here. And right where it folds in right there, we have a thickening of cells, these early mesodermal cells, and they're starting to spread out and go to either side from that point of infolding. So here's our spreading mesoderm from our primitive pit, and then we have our hypoblast layer of cells extending across below that. So anyway, this is neural plate. Ectoderm, this is surface ectoderm. I'll abbreviate with ectoderm there. We have our spreading mesoderm. We have the beginning of our endoderm that will form our gut. Right in here. Right there was our primitive pit. Beginning of our mesoderm as that folds in. These tissues move in and spread out as mesodermal tissues. So we have our three germ layers finally from what was initially just a flat pancake of cells. That's separated into two layers, epiblasts and hypoblasts. From our epiblasts we get all of these tissues that form the top layer, and in that top layer we get the formation of primitive streak. Primitive streak forms a primitive pit and Henson's node, and it's at that point that we get the infolding of mesoderm. Mesoderm starts to spread out between the two layers, so we end up with this sandwiched layer of tissue. And neural plate starts to form in the very middle of that surface layer of the embryonic, should say the blastodisc in front of the Henson's node up there. In the next drawings, we're going to be seeing what happens in the formation of the neural tube, in the formation of lateral mesoderm, how it's separated out to form the coelom, the body cavity, and we'll start talking about how the gut develops. Okay, then back to where we were with our chicken embryo. Remember, it has a large embryonic disc. And in that embryonic disc, there was a primitive streak that extended forward that laid down the mesoderm and some of our endoderm. Beneath that, we had our layer of hypoblasts originally, and into that we've incorporated some of the spreading tissues from the infolding at the primitive pit. Anterior to the primitive streak right here in the primitive pit in Henson's node, in this region we have the formation of the beginnings of the neural groove here, neural plate back here. So anyway, we're developing the nervous system from the ectoderm up in that front region. And we had the formation of mesoderm, the spreading out from that original primitive pit to fill this zone between the ectoderm and the hypoblast layer out here. And right down the middle of that, right down where we have this groove, we're going to have the formation of notochordal tissue. So this is going to become notochord underneath the ectoderm. Remember, this is a sandwich, so on top we have the blue, in the middle we have the red, and below that we have the orange in this picture. Let's draw that in a cross section here now. So we're going to go through this very top part of our embryo, showing we have surface ectoderm, then we have this thickening of neural ectoderm, so there's our thin surface ectoderm. Here's our thick neural ectoderm right here, nervous tissue developing right here. Underneath of that we have the beginnings of our notochord right here. Lateral to our notochord we have what we call lateral mesoderm, and our lateral mesoderm starts to form two different layers, a surface layer and a deeper layer here in close to the body. We're going to get some other structures to form from the mesoderm right in here, this portion. But then we have a single layer above and a single layer below this cavity space on the side here. So most of our lateral mesoderm is divided into two layers. And below that we're going to have our endodermal tissues. We'll just label it all endoderm, even though some of it's derived from that old hypoblast layer of tissue we had in there. Now that upper layer derived from lateral mesoderm here is going to be what we call somatic mesoderm. And the layer below down here is what we call splanchnic mesoderm. So we have a somatic layer and a splanchnic layer, and between the two we have the coelom, or body cavity. Body cavity. Here's notochord. Notochord mesoderm. Here's our neural groove. From that initial layer of ectoderm forms a neural plate and then a neural groove. It'll eventually form the neural tube. So the next drawing we make is going to be an expansion of this, show it a little bit further along in development to show what happens with this tissue, especially right here, lateral to the notochord. All right, the next stage in the development here, we'll draw a little more developed neural groove. It's not very pretty. Let's try again. Okay, now the next stage in our development of our embryo, we'll draw a more developed neural groove. So this is neural ectoderm here. It's almost a neural tube at this stage. Beneath that is going to be a notochord right in here. And lateral to that we'll have our lateral mesoderm, and it's going to start to form a couple of different components in here between the notochord and the lateral somatic and splanchnic layers. So out here we'll have our somatic layer of mesoderm, then we'll have our splanchnic layer, and between the two we have our body cavity. This is going to be called a somite, and that's going to be called intermediate mesoderm. So here's our somite here, our intermediate mesoderm here, and then our somatic layer of mesoderm and our splanchnic layer of mesoderm, coelom space in between. So the important thing in this picture is just the separation of mesodermal layers. There will be, of course, still the lower layer of endoderm going across through here as well. Well, let's go ahead and label this. This is notochord. This is somite. This is intermediate mesoderm. Lateral to that, we have the somatic layer of mesoderm and splanchnic mesoderm. Those events, those positions are not going to change much for a while. We're going to close in the neural tube next. We're going to get some special neural crest tissues to form at the corners of the neural tube, and we're going to see a special folding of these layers out here at the edge. You're going to bend upward and make a covering over the entire embryo here. So now our embryo, let's see, we've got to put that fold in out here. Embryo kind of bulges up where the neural tube is, and then we have these lateral folds to either side. We've got the neural tube formed in here, and where the neural tube separated from the overlying ectoderm, we have neural crest tissue. So here's our neural tube, neural crest, and surface ectoderm right there. Beneath that, we have the mesodermal layer. The notochord here, somite going in here beside the notochord and up along the neural tube a little bit right there. Intermediate mesoderm in here, and then the somatic layer of mesoderm that follows that fold in the overlying ectoderm, and then another layer that's going to follow along adjacent to the endoderm. So all of this is happening on top of the yolk, above the yolk, in a very small area, that blastodisc we described earlier. So all this down here would be yolk, and here's the embryo forming on top of it. It's just a few millimeters in length at this stage of development. So the embryo itself is just going to come from this part. These tissues out this way and out this way will not become part of the final embryo. So we have the, let's see, we'll label right here, surface ectoderm. Surface ectoderm here, neural crest, ectoderm, and then neural tube. Ectoderm. So our three layers of ectoderm, or three parts of ectoderm we have at this stage. This layer of mesoderm that folds up right here, along with the overlying surface ectoderm, forms what we call a chorioamniotic fold. And that fold is going to continue up, and from each side is going to form a bubble-like wrapper over the top of the embryo. So that chorioamniotic fold consists of a layer of surface ectoderm and a layer of somatic mesoderm. And we have a name for those two layers together, making up a single kind of a portion to the embryo, a single layer that forms this fold right here. We call it a somatopleur. So those two layers together make up a somatopleur. We're going to see that both the chorionic membrane and the amniotic membrane result from that fold development. So we call that a somatopleur. This lower layer down here, where we have a layer of splanchnic mesoderm, and a layer of endoderm, those two together make up a splachnopleur. Splachnopleur membrane. So in addition to the splachnopleur and somatopleur layers that we can label here, we could also label the coelom. The coelom portion that's going to be included in the embryo right here is called intra-embryonic coelom. And that is found out here. This is going to be extra-embryonic coelom. In the next picture, we'll draw the chorioamniotic folds completely covering the embryo. It won't look a lot different otherwise. It'll have still the same material other than that. Okay, in this diagram, we're going to go ahead and label all the parts that we had before, as well as the new features that we see here. Notice that the embryo body now is kind of lifted up away from the yolk a little bit more, and as it grows, as we see continued growth of the body of the bird, in this case, this layer of endoderm along here is going to rise up into the body along with the growth of the body. The head part of this embryo is going to grow in one direction and bring with it that endoderm we see here, and it's going to form essentially a tube as the head grows forward, another tube where the tail grows backward from its point of origin. But anyway, we don't see that yet at this stage. So let's point out a few things here. First of all, there's this double layer of surface ectoderm and somatic mesoderm we have here that forms the chorionic membrane. Chorionic membrane out here, on the outer part of that fold of the somatopleur, and from the inner part of the fold of the somatopleur right here, we have the amniotic membrane. So we have an amniotic membrane next to a chorionic membrane. Inside of that amniotic membrane is this space around the embryo. This is amniotic cavity. Amniotic cavity. I'm going to use a green here to show the limits of the embryo. So this is what we call a lateral body fold, this kind of tuck in on either side. So our embryo proper comes from that part. Everything else is extra embryonic in there. So we've already seen surface ectoderm, we've seen the neural crest ectoderm, we've got the neural tube going on in here. We have somite mesoderm, we have intermediate mesoderm, we have the coelom with the somatic layer of mesoderm and the splanchnic layer of mesoderm. This double layer right here of splanchnopleural membrane right there, that is the yolk sac membrane. Yolk sac membrane, or simply yolk sac. Eventually, we're going to see the yolk sac separate from the rest of the organism. We're going to have our gut form from this material right here, that endoderm and the mesoderm that lies adjacent to it, that splanchnic layer of mesoderm adjacent to the gut. There are a total of 4 extra embryonic membranes. We see chorionic and amniotic membranes up here derived from the somatopleural layers. And then down here we see a yolk sac membrane made up of splanchnopleural membrane. There's another splanchnopleural membrane that forms off of the hindgut. We've already talked about how the foregut goes one direction and the tail, the head of the body, head fold, grows in one direction and the tail grows out the other direction from the point of origin of the embryonic disc. As it does so, toward the tail end, we're going to form another kind of membrane we call the allantois, or the allantoic membrane, off the back end of the embryo. But we can't see it in this section. We have to look at a longitudinal cut of the animal, and that's what we'll do next. Okay, in this illustration here, we have that covering over the whole embryo. This is the head of the embryo. This is the tail of the embryo. It's still connected to all the yolk and membrane layers down here across this span right there. That started out as being just a few millimeters in length, not very large at all. And the body's beginning to grow with a head fold to the front and a tail fold to the back, sometimes called a head process and tail process, but anyway, the head and all those structures growing away from this point of attachment, and the tail and these structures growing away from this point of attachment here. So we still have on the outside a chorion and an amnion and an amniotic cavity, right? Down here we have the surface ectoderm in blue around the outside. We have the neural tube in the hatched portion right there. So the brain is going to form here in the head, and this is going to be spinal cord. Below that you have the notochord going through here. Now this is down the middle of the body, so you won't see any somite or intermediate mesoderm from the lateral mesoderm here, but around these membranes you do see that in that cut section. Now down here is the yolk, and this is the yolk sac. Down here, so this is the yolk sac membrane. This portion of the gut that comes into the embryo and comes up with the development of the head right here, this is called foregut. Foregut, and this part back here is called hindgut. The part in the middle, still where we have this connection to the yolk across here, that's referred to as midgut. So we have foregut, midgut, and hindgut. And off of the hindgut, we have this extra sac that I mentioned right here. This extra membrane forms a little sac, a little tag of membrane that comes off the hindgut right here. And this is the allantoic membrane. The allantoic membrane, forming the wall of the allantois, this sac that comes out here. Now this connection around from the embryo to all these extra embryonic membranes right here is referred to as a body stalk. The body stalk, right there. So, right up here we're going to have the mouth form at this end. In the case of a bird, it'd be a vent or a cloacal opening would be back here at this end of the gut. We'll talk more later about what happens with the allantois and the yolk portion of the membrane, of the embryo. But anyway, we have all four of our membranes on here now. We've got our outer chorionic membrane, or chorion. We have our inner amniotic membrane. We have our amniotic cavity, we have our yolk down here. We've got the cavity of our allantoic structure out here off the hindgut. So anyway, the next picture we draw, we are going to include the heart and blood vessels in this section right through here. And we're going to begin with the heart developing right in this area of the, nope, not that side, sorry. In this area right here, the splanchnic mesoderm in that part of the body. Splanchnic mesoderm just underneath the developing pharynx where the pharynx is forming. Okay, a little bit larger diagram here of the same thing. We've got the head up this way, the tail back here. We've got the membranes that surround the embryo around the outside. The notochord in red through here, the brain and spinal cord cross-hatched. Here's our body stalk right here where the embryo is attached to everything else. And in this drawing, I haven't put in the red coloration for the mesoderm next to the splanchnopleural membranes or the somatopleural membranes out here next to the chorion and amnion. I haven't put in a red layer or around the allantoic sac or around the yolk sac here because I wanted to put in some blood vessels, and we're going to use red to indicate the location of the blood vessels. So the heart forms right in this area here. So this would be the heart, and from the heart there's a blood vessel that runs forward and then sends out branches that go around the pharynx, both on the left side and on the right side. So I'll just draw in a few of these here. And we call these aortic arches. They come from the ventral side of the pharynx, go around the pharynx, and then coalesce, come together on the dorsal side up here. And there's a blood vessel that they feed into, simply called the dorsal aorta. So here's our dorsal aorta extending down toward the tail. So the heart, there's usually a little bit of what we call a ventral aorta on the ventral side leading into the aortic arches and then into the dorsal aorta. Now from here, the blood, one of the major blood vessels that leaves the dorsal aorta goes out through the body stalk around the yolk down here. And this is called the vitelline artery. Vitelline artery. So the vitelline artery feeds blood to the yolk region. That's where you're going to get your nutrients. So blood is going to have to come back from the vitelline region and come into the backside of the heart. And there are two vitelline veins, and I won't draw the entire veins in here, but I'll put them in purple to indicate that they're veins that come into the backside of the heart. There's a left and right vitelline vein coming in here. Now there's also a pair of vessels, a left and right vessel that goes to the allantois from the back part of the aorta here. So anyway, we've got a blood supply going out around the allantois and the mesoderm surrounding the allantois back here. So we have two allantoic arteries, two vitelline arteries, two allantoic veins, but only one, I should say two vitelline veins, but only one allantoic vein. And here's the allantoic vein coming in right here, coming from the allantois. And I won't draw out all the branches of the allantois. So we have an allantoic vein coming in here, and then two vitelline veins coming into the back of the heart. And that's the simple vascular arrangement you see in the embryonic chicken. We'll see it in the embryonic human, and it's basically the pattern in fish as well. So we have the heart right here. We have three aortic arches going around our pharynx. We have our dorsal aorta here. We have vitelline arteries. That go to our yolk. We have allantoic arteries. That send blood to the allantois. We have an allantoic vein that brings the blood back, a single allantoic vein. And paired vitelline veins. No place to write it in. Here we go. Vitelline veins. So there is circulation to other parts of the developing body, but they're not quite as important or large as these vessels we see here. The blood to and from the yolk is the main blood circulatory arrangement at this stage of development. There's some blood going to the allantoic sac and coming back from it in there as well. But anyway, so we have the blood from the heart goes forward through a ventral aorta, through the aortic arches to a dorsal aorta, then out through these major vessels to the allantois and the yolk sac, and then coming back from the yolk sac, we have vitelline veins, and from the allantois we have the allantoic vein. The allantoic vein, and allantoic arteries and vein, in placental mammals will be called the umbilical arteries and veins. So allantoic and umbilical vessels will be the same thing, but in this case, this allantoic sac, we're going to call them allantoic arteries and allantoic veins for that reason. But that's the simple arrangement of circulation in the early embryo, and like I said, resembles very much what you see in a fish heart. We'll notice that when we look at the shark dissection. We'll observe basically that same circulatory pathway. This does not look very much different from a human structure when we are looking at the overall shape of the embryo, the major components, and the blood circulatory system.